This lecture will deal with the topic of microbial indicators. Indicator organisms turn out to be very important for the analysis and design of disinfection systems, and that's because the microbiology that we identify in municipal wastewater samples tends to be very complicated. So it's not practical for us to try to quantify all microorganisms that might exist in a wastewater sample. As a surrogate for that, we choose to use the concentration of one or more indicator organisms. Let's start by considering wastewater microbiology. As I mentioned a minute ago, there's a wide range of microorganisms that exist in wastewater samples, including viruses, bacteria, vegetative cells and spores will be included in those bacterial populations, protozoa, including their resting stages, yeasts, algae, and many more. Among these microorganisms, most are non-pathogenic, but some are pathogenic. It turns out that in general, it's impractical or impossible to measure the entire inventory of microorganisms that might exist in a wastewater sample. So as an alternative, we seek to identify one or more indicator organisms that can be used as indices of microbial quality. And generally, the concentration of viable or infective indicator organisms in the effluent that's released from a wastewater treatment plant will form the basis for design of the disinfection system. There are a number of characteristics that we could associate with what we might identify in an ideal indicator. As an aside, the word ideal should give you a clue that it's very unlikely that we're going to find a microorganism that will satisfy all of these characteristics. But nonetheless, it's useful to identify what those characteristics might be. They include that they need to be common to the undisinfected wastewater samples we'll work with. In other words, they need to be easy to find. They need to be non-pathogenic to humans, and that's because the assays that we use to quantify the viable or infective concentrations of these indicators will require some culture-based method, meaning we're going to need to grow them. You don't want to be growing pathogens in the lab unless you absolutely have to. The assays that we use to quantify the concentrations of these viable or infective indicator organisms should be simple, inexpensive, and rapid to allow quantification using these culture-based methods. The presence or absence of an indicator organism should correlate strongly with the presence or absence of the true pathogens. And then lastly, we expect the indicator organisms to be at least as resistant to the disinfectants as the pathogens themselves. In other words, they should provide a conservative response in terms of how the microbial community will respond when a disinfectant is applied. There are a number of common bacterial indicators that are applied. Among the most common are various groups of coliform bacteria. Those groups and how they relate to each other are illustrated in the image on the right side of this screen. Common groups of coliform bacteria that are used as indicators include total coliforms, fecal coliforms, and more specifically E. coli. For wastewater discharges to marine or brackish environments, Enterococcus are oftentimes identified as an appropriate bacterial indicator. The selection of an indicator needs to be consistent with the intended use of the discharged water. That is what, in part, motivates the distinction between Enterococcus and coliform bacteria in these various settings. As I've mentioned previously, but I want to reiterate, quantification of the concentration of viable or infective indicator organism should be done by a culture-based method. In the context of bacterial analyses, the two most commonly used methods to quantify these indicator bacteria are membrane filtration methods and the use of fermentation tubes, where the concentration of the viable indicator organism is going to be quantified using a most probable number technique. In recent years, there's been an emergence of non-bacterial indicators, most common of which are the bacteriophage. The microscopic image in the bottom center of the screen indicates a bacterial cell that's been infected by phage. Bacteriophage are viruses that have the ability to infect bacterial cells. They do not have the ability to infect human cells. So that provides a distinct advantage for us because people can't get ill as a result of culturing these bacteriophage. They tend to be abundant and more persistent in wastewater than bacteria. They're excellent indicators of viral contamination because they themselves are viruses. There are culture-based assays that are available to quantify the infective concentration of these phage. The most common of these culture-based methods is the plaque assay, an example of which is illustrated in the lower left. There's a petri dish here that has a bacterial lawn that has grown, and the dots that are showing up there are plaques that have formed by infective viruses, bacteriophage specifically, that have infected this bacterial lawn that was grown in the petri dish. They tend also to be more resistant to disinfectants than bacteria. Among the bacteriophages that are used are several different groups, including F-specific coliphages and somatic coliphages. They tend to be structurally similar to human pathogenic viruses, and their responses to disinfectants tend to be similar to those of human viruses. In fact, oftentimes they're conservative slightly relative to human pathogens. I should point out that the most important, perhaps, pathogenic virus that exists in municipal wastewaters is the human norovirus. It is responsible for a large fraction of microbial diseases that are associated with municipal wastewater, but it's very difficult to grow in the laboratory. So it's natural to seek a surrogate for the human norovirus, and it turns out that these groups of coliphages that are illustrated in the upper right, F-specific and somatic coliphages, work well as those conservative indicators against human norovirus. To summarize, there's a broad spectrum of microorganisms that exist in wastewater samples. Rather than try to quantify all of them or even a large fraction of them, we focus on the concentration of viable or infective indicator organisms as an index of microbial water quality. The common bacterial indicators include coliforms and the Enterococcus, and bacteriophage have been identified within the last decade as an alternative to bacterial indicators. Lastly, the effluent concentration of viable or infective indicator organisms oftentimes forms the basis for design of disinfection systems.